Most of the lab was looking at what are the mechanisms of synaptic plasticity in the LA that underlie hair conditioning. And at the time, we knew that LTP could be induced in the thalamic input pathway and cortical input pathway to the LA. And so I started working with Mark Weisskopf, who was a postdoc at the time, and he was using a pairing protocol to induce LTP in the LA. And this had been used in the cortex and the hippocampus, but the attractive thing about this protocol for inducing LTP in the LA was that it seemed to mimic what was happening behaviorally, which is that if you pair a weak CS with strong US, then that induces plasticity, so that now the CS produces a strong response in the LA neuron. So we mimic that with presynaptic stimulation of the thalamic input fibers, which was supposed to represent the CS, and depolarization of the postsynaptic neuron, which was supposed to represent the US. And the nice thing about this was that pairing these two stimuli induced LTP in an LA neuron, but either of them alone was not sufficient to induce LTP. And this kind of LTP was blocked by antagonists of voltage-gated calcium channels. And so that was that. So in black are control cells, and in orange are the cells in the presence of verapamil, which is the calcium channel antagonist. And all of these LTP studies were done in collaboration with either Glenn Schaaf or Serena Rodriguez. And what Glenn did was he then injected a verapamil into the LA prior to training, and we saw that it had no effect on short-term memory, but it impaired long-term memory, suggesting that these calcium channels are needed for long-term memory, which is lovely. But we all know that the NMDA receptor is important for learning and for LTP. And Serena had shown that blocking the NR2B subunit of the NMDA receptor blocks both short-term and long-term memory for the tone and contextual hair memory. But when you block NMDA receptors, the entire receptor, or just the NR2B subunit, it does not prevent LTP induced by this pairing protocol. So initially this was a problem. So then we switched to using a high frequency potentiation to induce LTP, and this produced, this 30 Hertz tetanus produced a long depolarization of the postsynaptic cell. And if you induce LTP this way in an LA neuron, it is blocked by APV or blocking the NR2B subunit. And what was nice about this was that we saw that NMDA receptors and calcium channels both contribute to LTP and fear memory, but in different ways, and suggested the cellular mechanisms underlying short-term and long-term memory are different. And what I really liked about this study was that rather than just saying blocking NMDA receptors blocks LTP, it was a little more sophisticated in that now we have these two different protocols, one that seems to tap mechanisms underlying short-term memory and one which seems to tap mechanisms underlying long-term memory. So then we could ask more sophisticated questions, which Serena's going to talk about in a second. You know, calcium entry through one type of channel versus another type of channel. What kinases does that do? Does calcium activate in those different situations? So then the model that we came up with was during conditioning, calcium enters through both of these channels and drives potentiation, and NMDA receptors are needed for short-term memory, and both NMDA receptors and calcium channels are needed for long-term memory. So I came to, how long was I in the lab? 6 years or 7 years? And I lived 10 minutes away, and at the time, and I thought that this neighborhood was the center of the universe. And I still think that. And the amygdala was the most important part of the brain. And I still think that. But for my postdoc, I decided to expand my horizons. And so I bought a car, and I started commuting to Newark, and I'm working in Timmy Puray's lab. And I looked at how the amygdala modulates memory consolidation in other regions of the brain, the perivinal and entorhinal cortex. And this involved chronic in vivo recordings in cats, recording from multiple areas of the brain, the rhino cortices, the amygdala, the MPSC. But I'm not going to talk about that, actually. Denis' lab was great. I learned a lot in his lab. And then in the last year of my postdoc, Seville Duvarcy and I did a project looking at the relationship between the amygdala and the BNST, the beta nucleus mystery terminalis. And this is work that I am now continuing at Barnard, where I think this is my second year at Barnard. So the idea is, how does BNST activity modulate fear conditioning? Which is part of a larger question of how fear and anxiety interact. Because we know that learning to fear, the fear learning is critical for survival, but one hallmark of a number of anxiety disorders is that people generalize their fear to stimuli or safe situations. So this is bad. And just a little background about the BNST. It's active during stressful responses. Stimulating the BNST produces stress, lesioning the BNST reduces anxiety. And the amygdala and the BNST are really heavily interconnected. So the BNST is incredibly complicated. I think it has 31 different subnuclei. But in general, the BLA sends a strong projection to the BNST. The BNST and the CE are very similar in terms of their cell types and their projections, including to the HBA axis. And so the idea is maybe the BNST is modulating more subtle aspects of fear conditioning, such as CS specificity or contextual fear conditioning. And actually, Greg Sullivan, when he was in Joe's lab, looked at the effect of BNST lesions on fear conditioning. And he saw that, if you lesion the CE, animals do not fear condition. But lesions of the BNST do not prevent fear conditioning to tone, or they don't prevent increase in blood cortisol levels. They do prevent contextual fear conditioning. So we used, so Seville and I used a differential fear conditioning paradigm. So we had a CS plus, which is paired with a shock, CS minus, which is not. And the two noises are very different. So the CS plus is a white noise, the CS minus is a 2 kilohertz tone. And then we tested for context and for tone pattern. And all animals learn the CS plus US association. Animals learn to fear the context. And in general, they freeze more to the CS plus than the CS minus. And what's interesting, and what has always interested me about behavioral experiments, is that when you plot the averages, it looks lovely, but it masks a high degree of variability. And especially if you look at the context, some animals freeze 90% of the time in the context, some animals freeze 0% of the time. Most animals, or all animals, unless they don't get shocked, learn the CS plus US association. But there's a large degree of variability to the CS minus. Some freeze 90% of the time, if you average the responses to the 3 tones. And some freeze not at all. So I think this is really interesting. What is the source of this variability? To look at this a little in more detail, we calculated a discrimination index, which is how much animals freeze to the CS plus versus the CS minus. So if you freeze equally to both, your discrimination score is 0. If you have a low discrimination score, it means you freeze a lot to the CS minus. Basically, you've generalized your fear to the CS minus. If you have a high discrimination score, you freeze more to the CS plus than the CS minus. And what happens is, if you look at all of these animals, you have this basically this bimodal distribution. And you can define a cutoff versus high, to define high versus low. The cutoff doesn't have to be, there's some jiggle room in here, it doesn't have to be the exact average. But basically, you have 2 groups of animals. And we asked if there were other measures of fear conditioning which correlate with this. So this is an example of a poor discriminator. See, he actually freezes more to the CS minus than the CS plus. He freezes a lot in the context. And on a test of anxiety in the elevated plus maze, he spends most of his time in closed arms. This is an animal that freezes more to the CS plus than the CS minus. He doesn't freeze in the context, and he spends more time in the open arms. And these measures are correlated with each other. So we characterized the animals that didn't discriminate very well as having the anxious phenotype. They froze a lot in the context. They spent most of their time with the closed arms of the elevated clusters. And actually, they're also, anxious animals also extinguish slower. And you can define that as freezing to the first 3 tones versus middle tones. And this rate of extinction is also correlated with their ability to discriminate. What's important to realize is that they all freeze the same amount of CS+. That's the same. It's these other measures that are affected. So what happens? So what happens if you lesion the BNST? BNST lesion animals are in red. So you can already see what's going to happen. The average amount of freezing in the context is lower, and the variability has been reduced. Lesioning, as Greg Sullivan showed, doesn't affect freezing to the CS+, but there's a reduced variability of freezing to the CS-. Animals don't freeze as much to the CS-. And in fact, all but one of the lesioned animals have very high discrimination values. And these animals look like the subset of control animals that discriminate between the CS+ and CS-. So you have 2 groups of animals. One has the anxious phenotype and one that does not. And lesioning the BNST leaves you with good discriminators, produces good discriminators, low freezing to the CS-, low contextual freezing, low anxiety. And so this suggests that in normal subjects who display the anxious phenotype, BNST is more active, either baseline activity or sensor-driven activity. So the experiments that I'm currently doing at Barnard are looking at the mechanisms of this. And so there were a lot of people that contributed to, I only talked about a little bit of what I did in Joe's lab, because we only have 15 minutes, but a lot of people contributed to that work. And then Sybil and I did the work at Rutgers, and then I actually have a lab at Barnard, which surprises me. And there they are. So, thanks.
Thanks, Liz. For a while, I thought my whole lab was going to move to Rutgers.
There were
At least 3.
Wow.
Yeah, John, Sybil, and
Yeah.
you.
Yeah.
So it's looking bad for a while. But so a lot of the, I think your data are also consistent with this in terms of the BNST being particularly important for context. Is that?
Yeah, it is.
So the effect on the discrimination, you think, is modulated by the context?
No, I don't think so. I think that, so what I think is happening is that even though I've lumped all of these things together, context and CS specificity, I think they might actually be mediated differently. So I think it's an effect on the expression of fear in the context, and I think the BNST is affecting on the day of acquisition, and I think it's affecting how
These are pre-training lesions. Pre-training lesions.
Yeah. But now that we're dissecting how it looks like it's several different effects.
Okay.
Did you get an effect on extinction with the BNST lesions? Because you said they were poor extinguishers.
No. That's data that is new at Barnard, and we didn't look at lesions on that. It was just the control animals that correlated with that. So I don't have lesions on that.
So you said there are 31 sub-nuclei. How fine were your lesions?
No, I wish that we had more lesions or been able to correlate the histology with that, but